Assalamu alaikum. It's my pleasure to introduce Professor Moustafa El-Araby. Professor Moustafa El-Araby is Professor of Organic Pharmaceutical Chemistry at Helwan University. Now he is Professor of Medicinal Chemistry at King Abdulaziz University. Professor El-Araby got a Ph.D. from University of New York, USA. Also, he worked at industrial discovery of drug for 4 years in New Jersey. Also, he has many research focusing on antiviral and anti-cancer hemoglobin modulator. With his research with 6 patents and 27 publications. Dr. Moustafa El-Araby collaborate with Center Information of Virginia, also with other Helwan HSBR and many other international universities. Dr. Moustafa El-Araby will talk about deciphering NS4A binding as a new strategy to combat hepatitis C virus. طبعا انا احب بس اقول ان انا اول مره اسمع عن الاستراتيجي دي، يعني انا معلوماتي وهعرف بقى من الدكتور مصطفى انه related NS4A related with dengue virus. فعايزه اشوف الاستراتيجي دي، ومرحبا بيك من خلال مؤتمر كليه الصيدله جامعه حلوان.
Thanks, Dr. Professor Wafa Zaghari. It's my honor to speak to my home school. It is a big honor. I was invited to this conference. I hope you hear me first. Everyone hears me?
Yes.
Okay, just to make sure. It is. I hope you will enjoy this new approach to develop antiviral drugs. So allow me now to share my screen. Can you see the presentation, please?
Yes.
Okay. As you mentioned that deciphering NS4A as binding as a new concept for development of hepatitis C virus inhibitors was not really something, it is really rare in the literature, and this is for a reason. I wish to move to next slide to tell you that that hepatitis C virus is one of the flaviviridae viruses, and flaviviridae viruses include very dangerous viruses, just as you mentioned, Professor Wafa. Dengue virus is one of them, West Nile virus, Japanese fever, like some very, very dangerous viruses. But most of RNA viruses just have a little or a small genome that translate into really limited proteome. So here is the opportunity that RNA viruses, including, by the way, SARS-CoV-2 and all the SARS family of viruses, they just depend on limited number of proteins for growth and replication, and this constitutes an opportunity for researchers to develop antiviral drugs. We started this research actually 20 years ago or more, before any kind of thought like any drug for hepatitis C appeared in the market. It was the idea that to utilize the NS4A as a target. What is NS4A? NS4A is one of the smallest peptides that expressed in the proteome of the hepatitis C virus. It is 54 amino acids, and actually NS4A is also expressed among all flaviviridae viruses. But maybe they have different different rules in the virus growth. NS4A is important as a cofactor for activation of NS3 protein. NS3 protein has two functions. The protease and the N terminal is protease and the C terminal is helicase. Integration of NS4A is also important for integration of the viral protein to the host cell endoplasmic reticulum. NS4A also deflect NS3-4A complex also is important for deflection of host cell immune response, and this is very important for the pathogenicity of the hepatitis C virus. Structurally, we can see that the NS3 protein, this white sticks projecting from the protein structure are the catalytic triad that everybody or most of the research were directed to develop inhibitors that bind to the substrate site, inhibit the process of binding of the substrate. We were thinking differently. Here we thought about NS4A where it binds. NS4A it binds as a sandwich in between two beta sheets, A0 and the A1 of the NS3. And it is extremely important for activation of this enzyme. Once the 4A binds, the catalytic activity of the NS3 goes from 1 to 900. It increases almost 1,000 times. Here is a sequence of the NS4A for hepatitis C virus. It is different little bit between the genotypes of the viruses, but we focused on the genotype 4, which is common in the Middle East, especially in Egypt. And we find this little surprise, and also it was a pleasant surprise that studies conducted in the 90s discovered or revealed that only the central part of the NS4A is important to catalyze NS3 proteolytic activity, and they are sufficient. Moreover, and which is even more interesting, that one good study, also in 1996, asserted that not all the residues from the sequence 21 to 32 are important for activity. Residues, which are the 23, 25, 27, 29, and 31. Here is the first crystal structure appeared for the NS3-4A complex back in 1996 by Vertex researchers, and illustrates this point that the odd number residues are completely embedded under the surface of the protein. And this is very important. Then if you design something to interfere with the function of the 4A, you focus on the odd number residues because it's gonna bind well in deep pockets. And there is a reason for this because the beta sheet, the 4A is just bind as a beta sheet, and it has two faces. One is facing the A0, which is the external beta sheet, the external loop of the enzyme, and pushing on A0 will not change the catalytic or the structure of the apoprotein, and it wouldn't affect the catalytic activity, but interfering with the internal residues, the odd number residues, 23, 25, and 29. Why we are ignoring for now the glycine at 27? This is I will mention later in my presentation. But just remember that 27 is not like a really good candidate for our interference with the function of NS4A. And for good luck and bad luck, everyone focused on development of competitive substrate inhibitors, substrate site inhibitors for NS3/4A protease. No one and other proteins and combinations of them and have been really successful. However, no one thought about NS4A. Why we are thinking about 4A? Because as Professor Wafa said, it is common in many viruses. It is new target. If you have resistance, you have an arsenal of drugs that can overcome this problem. Everybody still hear me? It's just really quiet. Dr. Mu'taz?
Yep, Doctor.
Oh, okay. I'm just making sure that I'm not lost.
Not everything's fine. They're
okay.
Just trading, so.
Okay. Thanks very much. Okay. So why not we develop NS4A competitors and inhibit the function of the NS3? Actually, there was an interesting debate back in 1990s between two opinions. One from Vertex researchers appeared in their crystal structure article in Cell Journal in 1996, and they stated that the contact surface between NS3 and 4A is quite extensive and provides another possible site for the design of hepatitis C virus chemotherapeutic agent. While a very in hepatitis C, scientists based on the same reason, and he stated that Rafael de Francisco stated that the interaction between NS3 and 4A involves a very large surface area, and therefore it is not likely target for development of inhibitors, which is really puzzling to us. So it just took, we wanted to resolve this debate. First of all, just a sketch diagram for what really happened. If this is a APO enzyme, this is a substrate active site, and this is 4A. 4A is a 54 amino acid. The virus prepared separately, then it binds, organize active site, bind the substrate, and then go ahead with catalytic proteolysis and make a separation processing of the viral proteome. Our idea is very simple. It is just if you bring a bulkier synthetic peptide of 4A, especially the central region, and you just a mutant with something to the 4A site is gonna make a little changes in the conformation of the APO enzyme, but huge change in the catalytic activity, because our hypothesis is to corrupt the active site, making it unable to bind to the substrate, unable to function as a proteolytic enzyme. But beware of the experiment, because no one researched in this area before. I am a synthetic chemist, as Dr. Wafa said. I'm in the organic chemistry department. So our understanding, our ability and skills in this area is somewhat limited. However, with collaboration, with important collaboration, especially with Professor Samah Surur and Professor Stephen Arul, I will mention them later. But we could get help, develop, undertake the job to develop an assay for our purpose, because this assay is really is a warning sign. It is not a tricky, because smaller synthetic peptides that don't bind to the protein also form don't form a complex, don't form anything, but the APO enzyme by itself without 4A is inactive. So imagine that if you bind it is inactive, if you don't bind it is inactive too. So be aware of that, and we had to develop a suitable assay for our conditions. And so the objectives of this phase of the project was to answer important question: do NS4A synthetic analog bind or don't bind? If yes, it lead to inactive form of the enzyme, and if yes, NS4A pocket validated as a site for inhibition of the NS3 enzyme. Design was based on titration up of one site mutation on the positions 23, 25, and 29. As I said, the odd number residues only. And the titration in size, we have in this site, for example, there was a valine mutated to alanine. This is smaller size. However, we titrated up to cyclohexylglycine, which is a little bit bigger than isopropyl, then a natural residue, which is homoisoleucine, a little bit bigger or larger size, tryptophan and phenylalanine. Those are bulkier and aromatic residue. And we repeated the same thing for position 25 and 29. First of all, as I said, we had to address the binding issue, and it took us a big deal of time to develop assay. We cannot say if those peptides inhibit or inhibit, because we were like really asking the question in our way as people did in 1990s. We have to develop, and we are so many struggle. We would see that peptide number 50, which is a mutating of valine 23 to cyclohexylglycine 23, is the best one. Why is the best one? Because the NS4A using unlabeled technique. This is just to confirm you that crude data of the native 4A, the peptide binds higher and better using this technique. On all conditions and the time, we have the same good results within a really small margin of error. Here is another binding assay, but it was very important for our purpose because this is called the fluorescence anisotropy assay. First of all, it is labeled assay. We fluorescent label the NS4A, use it, and measure how it binds. Increase of the fluorescence indicate how it binds to the apoprotein, the NS3. And here is a trick. We were really fooled by what is in the literature. All the literature we spend, and you can ask Professor Surur, we spend about almost a year trying to reproduce what is in literature and what is in the catalog, the catalog or the kit catalog. And we find that in incubation, as on their direction, in incubation of the NS4A with NS3 for 15 minutes, as stated in all literature, doesn't produce that much fluorescence. We have to do it over at least 30 minutes. 45 minutes is the best fluorescence production, and it continue for more than 2 hours. You can continue seeing the fluorescence stable. And through this assay, we determined that the association, the affinity of the wild or the viral NS4A to NS3 is around 170 nanomolar. This assay helped us to measure if the peptide, the synthetic analog of NS4A from its site of binding. That was achieved really well using NS fluorescence anisotropy technique, because if you mix the NS4A, the ratio of the affinity, the KD affinity, and then start to add titrate up the amount of the competitor, the BIF15, then we'll see a decrease in the fluorescence, and it was really stable over the time. Either you measure it after 90 minutes, 120 minutes, determine that the affinity, the competition, the BIF15 really just compete and replace NS4A from its site of binding at affinity, KD, the association constant 70 nanomolar. Okay, we know that BIF15 is the best one, and it just achieved our hypothesis that the NS4A with only small change can bring large inhibition of the catalytic activity of NS3. Now we had like really a difficulty that up till now is still a difficulty, which is measuring the inhibition of the NS3 by this NS4A analogues. We find that mixing BIF15 with NS3 for 60 minutes, before 60 minutes doesn't inhibit the enzyme catalytic activity, but after 60 minutes, we find a big drop in the catalytic activity of the enzyme upon mixing of our synthetic peptide, while the 4A, the absence of 4A limited catalytic activity. 4A mixed with the enzyme, of course, it is a control, gives you 100% of activity. However, because we are using in this assay large amount of the enzyme itself, so adding a little bit of 4A, it reverses the inhibition and the enzyme reactivated again. And this is, I wanna assert that this is appears related, not related to the binding, because it was confirmed by 2 techniques, but it was the assay condition. We couldn't develop a better assay condition, but by using a large amount of the enzyme, and we are trying to fix it in the near future. Unfortunately, we couldn't conduct molecular structural biology studies by crystallography until now, but we ran molecular modeling by molecular dynamics by docking the BIF15 into the NS3, NS4A binding site, and we found that the complex reached stability after short time. And what was interesting here, that the cyclohexyl group didn't bring that much change in the loop next to it. The residue moved a little bit around it. So what happened really was in the catalytic trial. Before binding, the histidine 57 established a strong hydrogen bonding with serine 193 with optimum distance of between them. However, after molecular dynamics simulation, the distance, the serine 139 moved. I'm sorry, it should be the blue one is 4 before, 4 angstrom. After it changed to 8.4. I'm sorry for this mistake. It is after and before. I'm sorry. And this it moved away and became unable to establish any contact between serine 139 and histidine 57, and this is a reason why the enzyme lost its catalytic activity. And we are trying now to collaborate with someone in the United States to develop a crystal structure of BIF15 and NS3 to confirm this point. I wish to, if I have a little bit more time, to give you the one big step towards the development of non-peptide NS4A analogues. First, before I start that, the conclusion is simple. The conclusion is simple, that the NS4A peptide analogues can actually allosterically inhibit the function of the hepatitis C virus NS3 protein. Professor Wafa, do I have more time, or should I finish at this point? Would you please open the microphone? Yes.
Okay. You can hear now?
Yes, now, yeah.
Thank you so much, Dr. El-Araby. Now, questions from audience. Do you like to add some slides?
Yeah, there are some other slides. If you like to, if I have a little bit more time, I can.
Possible 5 minutes.
5 minutes?
Yes.
Okay. Okay. Let me tell you the most important thing, the de novo design of the non-peptide analogues, because this is very important for drug discovery, because peptides, after all, are not good drugs. We studying the structure of the bound with kinky area planar area on 4A, and we see they can be transformed into heterocyclic nucleus. First of all, we developed 1,7-naphthalidine scaffold, and we tried to synthesize those those compounds. Before we do that, we conducted the docking study. That was really old, one of the very old ideas. I had it like 20 years ago. However, the synthesis, the reaction of the aniline or the aromatic amines with glyoxal and the alkenes turned to be extremely challenging. It didn't work. Even it worked really with primitive analogues. The yield was really not really good using Bovarov reaction, and moreover, what was shocking, that the compounds were unstable 24 hours. By LC mass, you find no compound. However, the upside is what discovered that the reaction of glyoxals with aniline give extremely interesting pathway of reaction, because we published this paper very recently. We could resolve the structure. It is multi-substituted piperazine. However, we turned away from the naphthalidine. We designed the imidazole for this part, imidazole 2,5 dicarboxamides, and we found that some of them could bind well, although they are really much smaller than the peptide. Still, they can by competition assay, we could determine that the inhibition IC50 to 4A is reasonable at low micromolar ratio. And this is molecular dynamics study of the most potent very well. The 5 compounds, like this number 26, moves by molecular dynamics to protein and doesn't bind. And this primary, this is the literature so far in this project, and this is I'm proud of this team. They are were really dedicated. Of course, my best friend, he is the one who took the risk of doing this project with me, encouraged me with the in the first place to get the fund of it. Dr. Mahmoud El-Fay was dedicated to the biological screening at cows. Of course, our big scientist and consultant, Professor Samah Surur, he was really did extremely important effort facilitating all the biological screening, especially at cows, and all my colleagues, Dr. Man Khayat, Professor Hana Asfour, and Dr. Faida Bamana, and all students learned something from this project. Thank you very much. I am happy to listen to any question.
Thank you so much, my dear colleague, Professor El-Araby. Now, question from Dr. Samah. Now, some questions from audience. Dr. Samah Surur. دكتور سامح.
هو بعت السؤال ريتن فممكن نقراه.
طيب السؤال يا دكتور مصطفى ممكن تكتبه. What is your next step after proving the concept and identifying the potential inhibitor? الخطوة اللي بعد كده حضرتك هتعملها إن شاء الله. حضرتك إيه؟
Inshallah, first of all, we are moving to away from
some problem, Dr. Moustafa.
The presence of smaller peptides that are utilized by
Can you just
we are
Dr. Moustafa, please, Dr. Moustafa, please repeat the answer. We have certain problem in the connection. Can you please repeat the answer?
Hear me?
Yes, now we...
yes. You can hear me now?
Yes.
Okay. As I said, that hepatitis C is no longer an urgent problem in Egypt or anywhere. So we have the immediate thing, although we achieved everything, but it is really tempting to us to make the crystal structure of the at least the BIP15 with NS3 to prove the concept structurally. However, we are making plans to repeat or on the footstep of what we have achieved in this project to make similar approach to develop inhibitors of based on smaller peptide factors involved in the growth of other RNA viruses, such as dengue virus, such as SARS-CoV-2 virus, and it is in particular these two viruses are appealing to us.
What type of software you used for dynamic?
Okay, in the first study we had software that really disappeared from the world called Illexcor. Now are using Rodinger software package, and it is licensed to our university.
Do you expect advantage for this treatment over the conventional treatment?
Yes, of course, because any drug that is unknown to the virus will overcome any initial resistance that is developing right now against existing drugs. Now they moved from single or monotherapy to combination therapy. Combinations is still really working very well, but who guarantee that those combinations will still active after 20-25 years? And you know, Professor Wafa, that discovering a drug takes so long time. So now if we develop those drugs, compounds now, they might be in the market after 15 or 20 years. This is if we talk about hepatitis C virus. Talking about other viruses is just interfering with a function of small peptide factors is really appealing approach to us, and we are determined to pursue them in the future.
Do you have a lab for peptide synthesis in your university?
No. We purchased the peptide from an American company.
We will try to make a lab for peptide synthesis in Helwan University, inshallah.
Yes. It's my pleasure to do anything with Helwan. I work always with my Helwan colleagues, as you know.
One of my students take Ph.D. in Ohio and returned. We really think to begin this job.
Okay.
Any other questions? Thank you so much, Professor El-Araby.
Thank you very much.